G'day everyone, Ben the Spider Seeker here. Today I'm going to dive into what research in spiders looks like and where it might be heading. I mentioned in a previous video that my master's research showed that there's still a lot to learn about how funnel webs, as well as other spiders, are related to each other. But I'll focus on funnel webs today. There's so many undescribed species that are currently sitting in with other species that just need a little bit of extra work just to see if they're still the same species or if they are different. Just a heads up, where I can avoid jargon I have, but not every species has a common name, and I'll try to define any of the less common terms as I go. So I'll outline my experience and some of the interesting points from my master's research. I had limited experience in labs and fieldwork from my bachelor's degree, but nothing to the extent of what took me about 3 years to complete. In the end, I submitted a 66-page thesis with 9,066 words, which did not include the references or figures, and a total of about 13,500 words that I typed, with no AI, not even during the review of the gene sequencing. There was a total of about 38 figures containing images and descriptions and one nice big table. But that wasn't the hardest part of the whole experience. The first step, and possibly the most challenging part, was finding research supervisors. At first I reached out to ANU Emeritus Professor named Dave Roll. I have known Dave Roll since about 2015 when I completed the undergraduate invertebrate zoology course, for which he was the coordinator. Unfortunately, Dave Roll wasn't taking on extra students at the time, but they did steer me towards Professor Scott Keogh, who has an interest in molecular phylogenetics. I'll talk more about what that is in a bit. Professor Keogh also had a full load, but after a bit of a chat suggested that I check in with Dr. David Yates, the director of the National Insect Collection at CSIRO. David Yates agreed to help me out, and we discussed who else we would need on the team. I ended up with Dr. David Yates from the National Insect Collection at CSIRO in Canberra. I also ended up with Professor Dave Roll, who did agree to help me out and has given me so much of his time over the years to make sure that I know what I'm doing and support me in learning more. Dave accompanied me on several collection trips, sharing stories, resources and information about how to find certain species and other interesting bits and pieces to look out for. Dr. Andreas Zwick, also from CSIRO, is absolutely amazing when it comes to the technical aspects of the genetics and analysis that was required, and Professor Scott Keogh supported me directly as the formal supervisor through ANU, keeping me on pace with what I needed to be doing and when. I probably spent more than my share of my time in his office discussing plans and progress. Now, choosing the topic isn't really step 2, it's more of something to do with your potential supervisor in working out who needs to be brought in on the project. So it sort of happened at the same time. It was while I was talking to Dr. David Yates about potential projects. He asked what things interested me. Now, taxonomy in spiders and other invertebrates in Australia has largely been based on morphological characteristics, that is, what they look like. It's falling behind a lot of other groups where they've been using genetic information. We settled in on funnel webs and decided to investigate them using mitochondrial DNA, which is DNA stored within the mitochondria rather than the nucleus of the cell. While it's not as robust as nuclear DNA, mitochondrial genetic sequencing is significantly cheaper, quicker, easier, and perfect for a beginner in the lab. You might remember mitochondria as being the powerhouse of the cell. The nucleus stores the genetic material, though, that codes life. But mitochondrial is, as I said, cheaper, quicker, and easier. After deciding on this research path, Dr. Yates suggested I bring in Dr. Andreas Zwick, who was very experienced when it comes to mitochondrial genetic sequencing and did actually agree to help me out. So we had a project. Now to start work with a working project title. Something catchy, something thought-provoking, and something that would sell tickets. We thought of the perfect name. Molecular Systematics of Australian Funnel Web Spiders, Araneae, Myglomorphae, Atracidae. It is a mouthful, but the idea is to describe exactly what the project is and leave no questions about the point of the project. Molecular systematics is the process of using genetic information to build a phylogenetics tree, which is basically just a family tree. The focus is on Australian funnel-web spiders, with the scientific names afterwards being Araneae, which is the order that all spiders belong to. Mygalomorphae is a group called an infraorder, which is where the ancient or primitive spiders belong. And then, of course, Atracidae, which is the funnel-web family. Now, I mentioned this in my last video if you're keen on looking back and finding out a bit more about them. But before it all got started, the Advanced Master's degree at ANU is structured, or at least was when I did it, in such a way that the final thesis carries the grade for the course. In the build-up towards it, though, there's some milestones which have to be completed in order to progress through the course, even though they may not carry a grade. After finding the supervisors and working out the agreement, basically a contract of what I would do and what they would do, the initial seminar was first. The initial seminar was basically a public announcement of the plan of the project. Each honours and master's student had about exactly 20 minutes to present their project and be asked questions by the audience being made up of other masters and honours students and their supervisors, as well as a few interested academics who were available at the time, and even potential examiners that hadn't been announced yet. The initial seminar went quite well. There was lots of discussion beforehand with Dr. Andreas Zwick in particular to make sure that I understood the molecular methods being used and was able to answer some questions on it. During the seminar, I outlined the taxonomic history of funnel webs, basically what they've been called before, and it was not something I'd dived deeply into before, but that's something that I could probably do a separate video on, because that rabbit hole goes a little bit deeper than I was expecting. Once the seminar was done, all the masters and honours students were pretty supportive of each other, hanging out outside to talk about other projects, passions and challenges that we were expecting to face. Then, of course, it was time for something a bit more formal, something I wasn't used to. And that was the grant proposal. The grant proposal was probably one of the most challenging aspects of the project, not because it was intense, but it was because I was getting back into scientific writing after 7 years away from it, while I was also trying to wrap my head around the more technical aspects of the methods involved. It was that process in itself was intense. So my 4 research supervisors were incredibly helpful, providing feedback throughout the drafting process. Having the input of all of them made it so much easier. The proposal itself was about 1,500 words, and there was just 3 pages of references. So 3 pages of other research that I'd looked at. From there were a few other formalities like meeting with the examiners that had been assigned and regular check-ins regarding progress. The general structure of the Research Masters or Honours course was pretty broad though, and that allowed students basically the scope they needed to do their projects. The vast majority of my work during the first year was fieldwork, really, just collecting specimens. I would take time off work to travel or use my weekends when I was on shift, so I had conveniently a few days off between shifts. And I just used that time to drive around the countryside collecting from areas outside national parks. I would also go out actively on trips with Professor Dave Roll to collect specimens with permits in national parks and state forests. This involves some slightly hairy off-road driving at times. The Australian Museum in Sydney was kind enough to allow me access to their collection to borrow specimens for sequencing, and this is how I managed to get species that I could not find time to get in Queensland, Victoria and Tasmania. It was from the Australian Museum, though, that I found out another researcher was working on a similar project. I met with a few of the researchers on that project just to make sure that we had a different focus. My focus was largely on the Hadronychi genus, theirs was on the Atrax genus, and their work was later published in 2025, naming a few extra funnelweb species, which included the Newcastle funnelweb or Newcastle big boy, Atrax christenseni. In total, I managed to get a hold of 34 out of the 36 species that were described at the time. We also had an outgroup species, which I sampled from mouse spiders, the Actinopotidae family, and metallic tube spiders, the Pycnothelidae family. Only 22 species managed to get through the sequencing process, though, from a total of 114 specimens. None of the mouse spider sequences were successful, so the outgroup was the superficially very similar in appearance metallic tube spiders. The reason for some of the specimens not being successfully sequenced was related to the genetic degradation of the age of the specimens, with some of them collected from CSIRO and the Australian Museum were just decades old. So, in the field collections, I spent most of my time looking at basically a huge range of areas, which included dense, moist or mesic forest that was hard to get to. I ended up crawling up and down cliffs. We also ended up looking at private properties with permission, including underhouses, and destroying people's trees. They did, of course, help with the bobcat. And it's at this point that I would also like to acknowledge Scott Johnson. He's a good friend and a fellow funnelweb enthusiast. He goes by the Funnelweb Hunter on Facebook, where he collects Sydney funnelwebs for the anti-venom program and helps to educate so many people about them. I'll link his page in the description below. It was actually Scott who reached out to a few of his regular contacts about what I was doing and got us access to properties that had abundant funnelwebs. I also searched some of the drier areas with just enough humidity in the burrows to keep spiders from drying out. The burrows themselves varied a lot. Sometimes it was a matter of keeping a keen eye out for burrow entrances in tree trunks, or looking out for burrows under the base of trees or under rocks. And sometimes we would flip rocks and logs and dig carefully to follow a shallow or even very deep burrow at times. Then we might spot a burrow we weren't expecting in the top of a stump, leading to that little black dot. At the bottom there is Hadronychi cerborea. That's right near my thumb there. And we demolished that log by using pry bars, hatchets, and saws. So we finally basically burrowed ourselves down and found Hadronychi cerborea, that little southern tree-dwelling funnelweb there. Now, not every trip was successful though. I spent an entire day driving down to Wonboyne on the far south coast of New South Wales. I spent about 12 hours in total in that day driving to find the burrow entrance of a newly described species, Hadronychi nadji. I wasn't able to get it though because it was in a live tree, and digging it out would mean killing a large tree, and I wasn't particularly prepared for that. Now, after the collection, then the sampling, where I basically took little bits of the spider off and put them into little tubes. Preparing the lab samples, we sent them off for coding. Following the advice and methods of Dr. Andreas Swick and anyone else willing to help me, we walked through and eventually got back our sequences. And with some specialized software and adjustments, this is what they looked like. I'll be honest, understanding what was done for the sequencing is a stretch, so I won't be diving into that. But basically, I ended up with a bunch of genetic sequences, and then spent about 2 weeks trawling through the genomes, which I could change the color and grouping of using specialized software called AliView, A-L-I-View, and if anything stood out as remarkably different, I would use a search tool called BLAST, that is the Basic Local Alignment Search Tool, which is just a bunch of genome data stored online, and it is fully accessible. You just Google BLAST genetics and it usually comes up. If it came back as something other than a spider, I had to exclude that genetic data. So it sounds weird, but apparently I had a lot of mites, which did kind of make sense because a lot of spiders end up with a fairly heavy mite load on them. That is, they build up lots of mites. And what didn't make sense though was when I found a fish and bird genome that I ended up finding. I still can't work out how. Basically, I tidied it up, made sure it lined up, and eventually got it to a point where I could feed it into programs like FastTree and IQTree to generate the phylogeny. The family tree that shows how closely related all the species are. It's a bit busy though, so let's focus in a bit. Some specimens didn't line up with where the rest of their species were. This could indicate that there were a few different species and needed a bit more focus, or possibly a new species for description, or that I had stuffed up my identification. Now, for the most part, the genus groups held up, so Hadronychi was in itself a big, what they call monophyletic group. That is, they all came from one little branch. So this little branch here was where Hadronychi came from. The issue, though, was Illawarra, another genus, and let's focus a bit closer on them. So, Illawarra should only come from one branch, which would be monophyletic. But there are two groups of Illawarra. There's this orange group here, and that group is separate from the blue group here. The group with the blue arrow actually shares a branch with the Atrax genus. Now this could be because Illawarra is not a valid genus, being that it's paraphyletic they call it, which is split across different branches, or what seems more likely though, is that when I collected Illawarra from the one spot where I managed to find them, there were also some Atrax spiders mixed in, which were hard to identify as different since they were female and had the same burrow structure where I collected them. Now I did look at them fairly carefully and I was pretty happy calling them all Illawarra. Either way, this justifies a little bit more focus on Illawarra and Atrax and their genus division. As well as the Illawarra genus ending up with a question mark that needs further investigation, we also had Hadronychi caputorensis and Hadronychi formidabilis being in the same situation, where one population ended up looking like a different species. The Victorian species, Hadronychi jensenii and Hadronychi modesta, showed a bit of confusion too. These ones were from museum specimens though, and female, so it could have been misidentified in areas where their ranges are close together. The most heavily represented in my study was the species from my local area, Hadronychi visuta, the Blue Mountains funnelweb. Now what was interesting about them was this little guy from Owain. It turns out that the species from Owain is more closely related to Hadronychi monaro. And we can tell that because they have a little separate branch of their own. The most likely explanation in this case is that the Owain species is undescribed, but otherwise looks similar to Hadronychi visuta, just a little bit different. It did look a little bit weird, it just keyed out as Hadronychi visuta. The weird thing though, is that Hadronychi monaro is one of the alpine funnelwebs found about 200 kilometres from Owain. So it's further away than our Hadronychi visuta. The situation was the same with Hadronychi walkeri, looking like there might be a couple of species in there as well. Another thing of interest was that when Gray published the last big revision of Australian funnelwebs in 2010, he included species groups, that is, groups that he grouped together based on a few common morphological characteristics, like the bend in their second leg and that kind of thing. He did mention, though, that at least one of the groups was a group of convenience rather than taxonomic information. Unfortunately, my study didn't support those species groups. So that indicates that the groups might stand up to an even more thorough investigation into their genetics. And we can tell that because we've color-coded it here, and you've got blue within green and green within blue. What could happen, though, is that you instead look at rearranging the species groups based on the numbers here. So, for example, Hadronychi namula and the southern tree-dwelling funnelweb, Hadronychi cerborea, form group 5. Now, grouping should be monophyletic, which means they include all the branches called clades within that little group. Now, where each group divides would be where there was a common ancestor. It's not particularly the same as any of the species that are still around on that branch, but it's a point where the species divided and became separate. Going into how this works is an entire video in itself about evolutionary ecology, and I might save that for another time. Basically, for now, each group should be monophyletic, that is, all the branches are from one point. And if they're not, they would not be considered valid. The last and possibly most interesting part of building the phylogeny is finding some species that need work, possibly giving rise to undescribed species. We often call these new species, even though they've been around longer than we have. We just haven't named them yet. Hadronychi namula got their own video recently. I managed to collect a few from Gudgeonby and Wingelow, both very far from the original described location near Bateman's Bay. This is a massive range for funnelwebs. The two different populations I managed to get were also genetically distinct, that is, they're different. Now, it's a pretty clear indication that a more robust study would probably uncover a couple of undescribed species that are currently classed as Hadronychi namula. It's a similar situation to what happened with the Sydney funnel web, which was Atrax robustus and is now classed as 3 different species, including that Newcastle big boy that I mentioned. Hadronyche maracunda is in a similar situation. Now, I did struggle to find any of these until after the study, and even then I've only actually found one. A population near Crookle was highlighted to me at a show by someone who did road work in the area and found a large black spider. He conveniently got a photo to show me for confirmation, and he pinpointed where that road work was. Unfortunately though, the roadwork had managed to decimate the population, and despite finding the exact spot, I wasn't actually able to find any spiders. This happened more than I would like. Either way, Hadronyche maracunda deserves a bit of attention too. So, I finished the research, found out that Australian funnel-web spiders need a lot of work combining morphological and more robust genetic work to uncover just how many different species there are, and why there is so much variation within and between species. There is interest in this further research, with at least one researcher having already reached out to me to use my thesis in their own research, furthering our understanding of funnel-web spiders and likely resulting in more species being described. For spiders rated as some of the most toxic in the world, understanding their history is important to work out where their potent venom has come from and what spiders might have it. For now, I'll be watching this space with a keen interest, helping out where I can, and I'll share what I find out. A big thank you to my supervisors, family and friends who supported me on this journey, and of course, thanks to you as well for watching. It's been a long video, describing 3 years of my life studying spiders. Very proud to say that I now have a master's degree in spiders, and hope to continue studying and contributing to our understanding as well as bringing that knowledge to a wider audience. If you're enjoying this kind of thing, please subscribe, comment, like and share with any friends who might enjoy it as well. I do try to upload at least every couple of weeks. As I find a routine that works, I'll become a bit more regular with my uploads as well. If you have any questions, let me know in the comments. You never know, your answer might warrant its own video. For now, thanks again, and stay safe. Catch you later.